Hi everyone, my name is Eddie and I'm a senior here at the University of Arizona studying neuroscience and cognitive science. I'm standing right outside Gould Simpson, which is where my lab is located. I'll be giving you a short lab tour, telling you a little bit about myself, and then telling you about the undergraduate lab experience as a whole. I hope you enjoy. All right guys, welcome. So first of all, I just want to give a huge shout out to you. Welcome to the new students, and we're just so excited that you're here. We know that it's been a really difficult transition. With everything going on, we just want to thank you for being so flexible. And we're really excited that you're getting involved, whether it be virtually, in person, or a combination of both, whatever's most comfortable to you. We're just really excited that you're here, and we want to welcome you the best we can and support you the best we can. So with that said, please reach out to us if you have any questions. Please reach out to me. I'm happy to do any one-on-one Zoom calls or email back and forth about the student experience, what undergraduate research looks like, from the student side of things, and then what the College of Science looks like as a student. So with that said, welcome to the lab. We're a pretty small lab housed here in the Department of Neuroscience in the College of Science, and we are studying the vocal symptoms of Parkinson's disease. So Parkinson's disease is a neurodegenerative disorder, and one of the symptoms of Parkinson's is vocal symptoms. So people who have Parkinson's often have vocal deficits like horsey breath, reduced loudness in their voice, and reduced pitch. But we don't know a whole lot about what causes those vocal symptoms of Parkinson's. So because of that, we're using the zebra-finch songbird to understand what happens at the molecular level and at the behavioral level with the vocal symptoms of Parkinson's. So why zebra finches? Well, zebra finches have a really analogous vocal circuit for both vocal production and vocal modulation compared to humans. So males are actually the only zebra finches that can sing. And we can take advantage of the well-understood and homologous pathway to try and understand what happens in humans, what underlies the vocal changes in humans, and what can we see at the neuronal level, and can anything be done about it? Can we catch these symptoms early on before any of the other motor symptoms happen, like balance issues and coordination issues, and those sorts of things. So my role in the lab is to study a protein that's been linked to Parkinson's, and there's a mutation in the synuclein alpha gene that's basically been shown to cause Parkinson's disease. But we don't know a whole lot about what happens in the vocal symptoms. So I've been in the lab since I was a sophomore, and I'm currently a senior, so I started off my first semester sophomore year, and I've done a combination of both behavioral and molecular validations to try and understand what's happening. So what do I mean by both? Well, molecular validation happens right here in the lab. So that involves using tissue from our zebra finches and doing all sorts of different experiments on those to try and understand what type of proteins are involved in the vocal symptoms of Parkinson's, what type of neurons are being affected, those sorts of things. And then I've also done some behavioral analysis. So that's not done in the lab typically, that's done on a computer. So what that looks like for me is counting song. So I want to see how song in finches looks different before we've done any sort of manipulation to it and after. And by manipulation, I mean basically expose some subset of birds to some of the proteins or overexpressed proteins that we know have been linked to Parkinson's. So it's been pretty 50-50. I've done a good combination of both. And right now I'll be showing you a little bit about what happens in the wet lab or the molecular validation of things. So this bench right here is actually where I spend a lot of my time. And at this bench, I use a technique called immunohistochemistry. So immunohistochemistry is a lab technique that, it's very common across different molecular labs, and it's a very common molecular technique. And it allows us to visualize what proteins are being expressed in the brain in different tissues, and what type of cells are being expressed in, what type of neurons that are being expressed in, that sort of thing. So to do that, we use antibodies, and antibodies tag onto specific proteins, and then we use another antibody that allows us to visualize that. So basically, we then go onto a microscope once all that's done, and we're able to see the proteins being expressed in the tissue. So what I'm doing at this bench is basically getting the tissue prepared, doing several washes, cleaning the tissue, getting the slides prepared to allow for the antibodies to bind onto the protein. And once that's done, I take the slides and I go to an imaging core, which is actually not found here in the lab, but it's a shared collaborative space with different microscopes that all the labs can use here on campus. And I then I image those slides, and that's how I get my results. So I mentioned a core, and that's actually really common here in the College of Science, not just in biological labs, but all labs. So cores are just shared spaces that all labs can use, and that's the one thing about research here at the U of A. It's very collaborative, so it's not just one person, one professor working on one research project. Oftentimes professors will collaborate on different projects, and that's the same thing with students. So students can collaborate with one another, and we're very much about sharing our work and sharing our spaces and what can we do together. So anyway, what's done a lot, at this bench is getting all that prepared, and it's really cool to come in on my own time. I have deadlines that I have to do things by, but I'm never told, I have to come in from, 8 to noon on this day. Really, it's about me coming in on my own time, around classes and things. And that's the one thing about research is professors know that you're a student first. And so, I go to school first, I get my academic things done, and then after that, I can come into the lab and get all these experiments done, which is really cool. So I'll take you through some more parts of the lab, but this is kind of where I spend a lot of my own time. Some of the other students that were involved in other projects or in other aspects of the project and use different techniques, and I'll be taking you a little bit through that as well. Okay, so the first thing you'll see here on your left is something called a cryostat. So in order for us to be able to visualize any of the proteins that we're trying to study in our project, the brain has to be sliced really, really thin, and that's what a cryostat does. So a cryostat is basically a really cold machine. You'll see right now it's actually kept at negative 15 degrees Celsius. And the brain can mount onto that thing right there, the round thing right there. And we can slice the brain really, really thin at cold conditions. And basically, once it's sliced, we can then mount it onto microscope slides, and after that, we can do the different immunohistochemistry experiments that I told you about. So it has to be kept really cold because we don't want the brain to fall apart, we don't want the brain to crumble, and that can happen if it's kept too warm. So there's a perfect balance, there's a perfect temperature where the brain can be sliced, and that's exactly why it's kept at negative 15 degrees. So that's kind of what a cryostat is, and I spend a lot of my time here too, just getting our brains prepared and getting our tissue prepared. That's what our cryostat allows us to do. So you'll see just some safety things right there. We have our fume hood. We spend a lot of time in there. A lot of these chemicals you don't want to be smelling directly, so we'll work a lot of our time down there. We keep some of our reagents up there, and we have our freezer right here. So I talked earlier about using antibodies that allows us to visualize the protein, and that all of our antibodies have to be kept at a perfect temperature. So that's why we have a minus 20 degree freezer. So this right here is our RNA isolation station. So RNA comes right after DNA in the central dogma, and it's one of the ways or one of the techniques that you can see what genes are being expressed in tissue. And what we do is we isolate the RNA here at this bench, and then we send it off to a core where they do the sequencing. Another way of looking to see what genes are being expressed is through an experiment called RT-PCR, which is done with DNA. Now, RT-PCR is a really common technique, and one that's come up a lot right now with COVID-19. So one of the ways they diagnose people who have COVID-19 is through an RT-PCR experiment, where they swab the inside of your nose or the back of your throat to try and pick up some of those viral cells. They then run an RT-PCR experiment to amplify the DNA, and if that DNA matches the DNA of the known virus that causes COVID-19, you test positive. And if it doesn't, you test negative. We're not doing any sort of diagnostic experiments here or any sort of COVID-19 research, but I provided that analogy so you could see what sort of experiments we're running. We're trying to see whether the RNA that we're sequencing, that's being sent off to be sequenced, matches what we expect to see in tissue. And if it doesn't, it means something's being upregulated or downregulated. So Western blotting is often done in this part of the lab, and Western blotting is another technique that's used to see how much protein is found in different tissue. As you can see, it takes a whole lot of different reagents and different tools and things to be able to get the tissue prepared, and so we often keep stocked up in here. Okay, so like I said earlier, I've been in the lab since I was a sophomore, and I'm now a senior, so it's been about 2 years. And I joined the lab because my major sent out an announcement to all of our students saying, there's a graduate student in the lab who's looking for an undergrad. If you're interested, just send a short email and, they'll take it from there. And that's exactly what I did. So I interviewed with the graduate student and then with Dr. Miller, and they both liked me enough, who knows why, but they liked me enough to choose me to be a part of their lab, and that's how I'm here. And that's one way that different professors will basically recruit for undergraduate students. So my biggest advice is to constantly check your email, check in with your advising team, check your advising team's emails or any announcements that the college or major might send out, because there's going to be a lot of different labs recruiting for students that way. My second advice, and this is one that a lot of my friends have done and different classmates have done, and that's worked really well for them, is go on your department website, so whatever that might be, whether it's geosciences or physics or astronomy, neuroscience, biology, MCB, computer science, the list goes on. Go on your department website and look at professors that you're genuinely interested in. And look through some of their research, look at what they've done, and send them a really short email. Remember that professors get so many emails throughout the day, and you can imagine how much that's been amplified with COVID-19. So send them a short email and basically say, hey, I'm looking to be in your lab. I'm a student, in so-and-so major, and I'm really interested in the work that you do. I would actually even advise you to look at some of their papers. So see what they've already published, take a quick look at their method, at their introduction, at their abstract, see what they've done. And if you're interested in that, tell them. It should be a really short email and basically send it off to them. And send it to as many professors as you're interested in. Some of them won't respond, and don't take that personally. Honestly, follow up with them. They're, like I said, they're really busy, and sometimes emails get buried. So follow up with them, do whatever you need to do, and eventually I guarantee that you'll find a research position here at the university. We are a very, very undergraduate-focused university when it comes to research, and any undergrad who wants to get involved in research will eventually be able to, whether that's your freshman year or later on, you're absolutely able to do that and capable of doing that. So in addition to being in the lab, I've also been a part of different undergraduate paid experiences as well. So throughout the semester, I get credit for being in the lab, but I've also gotten paid for it through the undergraduate biology research program and through a program called Blazer, which I've just finished this summer. So the undergraduate biology research program, which stands for UBERP, you can go on their website, uberp.arizona.edu, and they're through the molecular and cellular biology department, but you can actually get paired up with a mentor, a faculty mentor, and work in their lab full-time over the summer. You also spend a lot of the time going to different talks and hearing different professors talk about their own research. It's a great program. There's other programs like MARC and UROC, and you can do research with them, and they help with a lot of grad school prep. You get paid to be a part of their program, so that's one of the biggest things is you can actually get paid even as an undergrad. And then I just did a program called Blazer this summer, and that helped a lot with a lot of pre-med prep as well. So there's a lot of different opportunities. I just talked about the biological sciences, but there's so many out in astronomy. I know that there's a lot of different NASA grants. I've had a lot of friends who've had a lot of success with that in space sciences and physics, so it's been a really great part of being an undergrad, and that's something that they can highlight both for graduate schools and in biological sciences for grad school and med school, those sorts of things. So it's been really great to be able to be involved, and undergrads can heavily be involved. Like I said, you can get paid and even get published. So if you do a lot of work, you contribute really heavily to a project, then you can absolutely be published as an undergrad, and I'm hoping to be able to do that. I've had a lot of friends and different classmates who've been able to do that, so it's really not uncommon for you to do that. It's really about getting involved and how can you, find research that you're genuinely interested in. You're not just doing it, as a resume builder, but it's really great to be able to be in a lab that's supportive and to be with a faculty member who cares about me and who supports me through my academic and personal journeys as well. So thank you so much for tuning into this lab tour. I'm, really excited that you were able to listen, and I know I talked a lot, but if you have any questions, please reach out to me. I will literally do anything that I can to meet with you one-on-one, via Zoom or email back and forth, whatever it takes. We'll work something out. And, I just want to give you our most sincere welcome to the U of A and welcome to the College of Science. We're really excited that you're here.